This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 314, recorded on December 12th, 2014. Hey everybody, I'm Vincent Racaniello, and you are listening to TWiV, the podcast all about viruses. Joining me today from southeastern Michigan, Kathy Spindler.
Hi, everybody.
How are you?
Fine. And the weather's great. It's a sunny day, and it's cold, but now I already forgot the temperatures that I told you.
5
36
degrees?
Fahrenheit, 2 degrees Celsius.
Yeah, it's 2 degrees here also. It's a nice sunny day, but it is chilly. But actually, winter isn't officially here yet, right?
Correct.
But this is winter weather.
Mm-hmm.
Also joining us today from Western Massachusetts, Alan Dove.
Good to be here.
How have you been, Alan?
Doing okay, and better now that this low pressure system that's just been parked pretty much in my yard all week is finally.
Wait, low pressure
out.
means what?
That's the bad weather system.
Bad
that's
weather?
The low barometric pressure. It gives you the usually it's associated with lots of moisture. It gives you the clouds, the precipitation. And this one usually they kind of blow through. This one just kind of blew up into New England and sat here.
Do you have snow?
We got snow and rain and fog and mist and more snow, and it didn't really stick to the roads. It was just nuisance weather all week and really glum.
We had a ton of rain last winter.
Now it's finally clearing up. Oh yeah, we got like
last
3
week
inches of rain.
Yeah, Friday and Saturday, and then also this week, and then it snowed a little. It's still really schizophrenic weather.
Finally clearing up. Supposed to be nice this weekend.
Also joining us today from North Central Florida, Rich Condit.
Hi, everybody.
Where the weather's nice, right?
65 degrees, 18 C, not a cloud in the sky. It's beautiful.
Yeah, 65 is not so great.
It is chilly by Florida standards, yes. I had to sort of put on 4 layers to bike in this morning.
4 layers.
But I'm not, I don't have to shovel this stuff.
No.
You don't, but you did in Buffalo, right?
That's true. I paid my dues. I didn't have a wobble, though.
You had your back, right?
Yeah, I had my back. No, I had money. I paid a guy. My brother tells me anything's possible if you're handy with money.
It is still the greatest labor-saving device.
My colleague lives up in Westchester, okay? And you know what he has under his driveway so he doesn't have to do anything?
Oh, he has one of those heating systems?
Yeah, he's got pipes full of hot water.
I would love one of those. It would cost as much as one of my cars. Both of my cars.
He's got a heater in the garage, and when it snows, he turns it on, and it never accumulates.
That's wonderful.
Isn't that amazing?
Wow.
That's what money will get you.
Yeah.
Well, a few weeks ago, they had a ton of snow in Buffalo, Rich, remember?
They did.
You probably were chuckling.
It's one of those classic lake effect storms where actually the city didn't get a whole lot, but the south towns got clobbered. Yeah.
I just got a text here from Paul Dupre. Huh? Is there any number for the downloads of Threading the Needle? Oh, yeah. The video? Sure. Video, I think 40,000.
Wow.
That's pretty good.
Yeah.
I wonder why there's so many.
Wow, there really isn't anything.
Because it's great. That's why. Come on.
Yeah, but you can do great stuff and nobody watches it. You know,
It went viral.
It did. All right, today we have some follow-up. Over on Facebook, after we did the 2 norovirus papers, someone wrote, didn't the previous noro paper you highlighted a couple weeks ago say noroviruses need bacteria for carbohydrates? Now this paper implies they can do without. Help! Can you explain, please?
Yeah, there was a little bit of tension between those two, wasn't there?
Tension. So I had, actually that Friday that we did the second paper, which showed that you can take nodobiotic mice and whose intestines are all screwed up, and norovirus infection fixes it. I had emailed Stephanie, and she gave us a reply, which we read last time. I had emailed Christiane Volbus, and she replied after the show. So here's what she said. My take on this question is that bacteria promote but are not absolutely required for virus infection. germ-free mice have M cells, and our data in vitro show that murine norovirus can be transcytosed by M cells without bacteria. Plus, murine norovirus can infect its target cells without bacterial help. So as long as the virus gets across the barrier, infection can start. Bacteria just make the process more efficient somehow. Yeah, so that makes perfect sense, right?
Yeah, I think that as time goes on, this is going to get more and more complicated. It's not a black and white story.
No.
Yeah.
Nothing is.
And transcytosis still blows my mind. I think I was not really aware of the whole concept of transcytosis until we encountered this it was some sort of paper using a viral protein that's ordinarily directs transcytosis in mosquitoes and a recombinant DNA technology to deliver something to the mosquitoes or something like that. At any rate, in my mind, a virus infects a cell and it's game over.
Yep.
All right? And everything changes and stuff. And the notion that this thing would be designed so that it would actually enter a cell and exit on the other side of this barrier and leave the cell probably otherwise okay just is crazy.
Yep.
Crazy.
But those barrier cells have to transport other things that way,
yeah,
too.
I guess. I mean, it's crazy to me, okay?
Yeah.
Doesn't mean it's crazy.
I am agreeing with you on that. But in retrospect, it kind of makes sense that there'd be some mechanism that would accomplish that.
Kathy, can you take Haley's?
Wait, but I was just going to say a comment on
Wait.
on why Christiana's reply might have been delayed. She's on sabbatical in Germany, and from my calculation, she's 6 hours ahead of us. So she might have already, been at home in the evening drinking wine or something by the time you sent your email.
Which is what I would have liked to have done.
Okay, so you want me to read
Haley.
We did Robin's already? Okay.
No, I'm sorry, we didn't. You're right.
No, we did.
All right.
You're
okay.
Robin.
Noroviruses and mice. Viruses, the kind that keep you well. With the shift in perspectives to microbiomes comes a paradigm shift. Koch's postulates will remain valid within their box, but now we have to deal with the whole world outside the box. Ecosystems are not only more complex than we think, but more complex than we can think. This quote from ecologist Frank Egler was probably based on a 1927 statement by evolutionary biologist J.B.S. Haldane, who said, "My suspicion is that the universe is not only queerer than we suppose, but queerer than we can suppose." Let us now try to revamp our understanding so that a person who is full of it may be a well-adjusted individual in possession of a valuable resource. And with reference to one of the TWiV podcasts, I can't remember which, a mouse with symptoms would be a most unusual mouse. It would be a talking mouse.
Yeah, we always do that. I'm sorry.
Yeah.
So what are the words? Symptoms and
Signs.
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 Signs. Okay.
Right.
That's another good title, Signs and Symptoms.
Yeah.
Okay, Alan Dove.
Haley writes, "Dear TWiV professors, your discussion about the human virome and finding potentially beneficial viruses got me thinking. For bacteria, scientists can take advantage of so-called universal primers to amplify DNA from all sorts of species that may be present in the sample, giving them a sort of ecological snapshot of what may be there, even if they can't culture it. Is there any method that we have now to take a general survey of what viruses might be present in the sample, even though they may not be causing any noticeable effects? I guess viruses are too varied to have universal primers, but couldn't you start by priming for virus families that have consensus sequences in some of their genomes, like where they code for their polymerase? At least then you could take a human herpes snapshot or a norovirus snapshot and thus piece together a partial view of the virome that way. Thanks so much for your podcasts."
All true.
Yes.
Right on the money.
Yeah, and in fact, that's how these viromics experiments are generally done. You use primers that are generic enough that you expect them to amplify large classes of viruses.
Yeah, but beyond that, you can't get everything, so you sequence.
Yeah, you do deep sequencing. So you do unbiased discovery, because you can look for families, but there are a lot of dark matter out there that won't bind to your primers, right?
Yeah, and the sequencing equipment is now sophisticated enough that you can take very, very mixed, almost crude samples and spit it in there and get sequence information. Of course, you get sequence information on everything that's in there, but then you can start to use the computers to dice out what the viruses are.
This actually comes up in one of our papers today, and they mention this specifically, that they can't, there's no generalized way of doing it.
Right. So the two omics approaches that people are using for these things are ribosomal RNA, which is the one you use for bacteria, and that's conserved enough that you can actually use primers for it and sequence just all the ribosomal RNAs of bacterial origin in a sample. And then there's the metagenomic approach, which is just sequence everything. And metagenomics is what you have to do for viruses.
I wonder what Haley's background is. I mean, the question implies that she's not, doing this stuff for a living, but the question also reveals that she's right on target.
Yes.
Yeah, the bacteria hunters are lucky. They can do 16S.
Right.
Pair up one or two pairs of primers, man. Wow.
That's too easy.
Rich Condit, you're next.
Peter writes, Dear Doctors TWiV, I have a question in response to your discussion of Peter's email read in TWiV 305. I don't doubt that norovirus can be transmitted via vomit. I'm just wondering whether replication in the stomach is required. I would find it remarkable if norovirus is able to replicate at stomach pH. I assume that only stomach contents and not the one of small intestine is ejected, even during violent vomiting of a typical norovirus infection. I love all the TWiX podcasts and listen to them mostly within 1 or 2 days of their release. Being 13 hours ahead in time of the U.S. East Coast, TWiV makes me looking forward to Mondays. I am a biopharmaceutical process engineer at my job. Viruses are something to inactivate and remove from the product, typically by low pH or a 20 nanometer filtration. Chromatography steps help too, even if it's not their primary purpose. This is just my second email to TWiV. The first one was years ago for the discussion of the Vesuvius contamination of Genzyme's manufacturing plant in Allston, Massachusetts. I remember that.
Yep.
It's 9 a.m. in Icheon and currently 15C under clear blue sky with a perfect Korean fall day, high 21C ahead of us. Regards from Icheon, Peter. So I wrote Stephanie about this, and Vincent wrote Kim Green. So Stephanie says, I agree, it seems unlikely that noroviruses replicate in the stomach. The most common idea I've heard regarding the pathophysiology of norovirus-induced vomiting is that the virus affects gastric or enteric motor functions. For example, people infected with a human norovirus and interferon knockout mice infected with murine norovirus both display delayed gastric emptying, meaning the contents of the stomach can't be emptied into the intestine, thus the stomach bloats. This could happen if motor functions were impaired, leading a person to vomit. And she also suggested that we pick vomiting Larry, but I told her it had been picked twice. But it's clear that virus is ejected during a vomiting episode. There was a presentation at ASV this year in the Khaleesi session where someone quantified virus in vomitus, and there was quite a lot of it. Not near as much as in feces, but quite a bit. As to how the virus accumulates in the stomach to begin with, I must admit that I haven't given this a lot of thought. I've heard from one researcher that a particular mouse norovirus strain replicates in the stomach, but I haven't seen the data, and it's unpublished and not the norm. I would guess for most, if not all strains, that it's backwash. Those were my words in my letter to her, from the small intestine. But why would that happen? Loosening of the pyloric junction between the stomach and the small intestine seems like peristalsis should be moving everything in the other direction. If you have a discussion on this and come up with some interesting ideas, I'm curious to know. Nice segue into Kim Green. Do you want to read that one, Vincent? Or do you want me to go on?
You can go on.
So Vincent wrote Kim Green, and she wrote back, interesting question. There are some who believe that noroviruses might indeed replicate in the stomach, and she gives a reference to that. I consulted with the gastroenterologist in my group. She said that normally vomiting generally expels stomach contents. However, with the heave of vomiting, the duodenum can contract, and the upper intestinal contents can be regurgitated. When vomit is yellow, been there, it is it is often the bile from the upper intestinal tract. Bingo.
TMI.
I have to look for that next time. I think we have another title, When Vomit is Yellow.
Yeah. But to me, that makes all kinds of sense.
I guess.
So that's the backwash hypothesis.
I was with Stephanie thinking that never happens. Why would he go backwards? But yeah, there you go. A gastroenterologist, they know, right?
I guess it's the titer in the small intestine is huge. So just a little bit of that is going to give a fair titer in vomit.
All right, we have one more from Robin. Killing gut bacteria killed the patient. Now, Robin is a physician. I had a patient who was on Coumadin, vitamin K antagonist. Vitamin K is needed by the liver in the synthesis of a number of proteins, including several factors in the blood coagulation cascade. Antagonizing vitamin K slows blood clotting and helps in conditions where excessive blood clotting is a problem. Two sources of vitamin K are green leafy vegetables and gut bacteria. Antibiotics, by disrupting gut bacteria, decrease available vitamin K and enhance the effect of Coumadin. Particularly egregious in this regard are the quinolones, such as Cipro. Well, this patient had been started on Cipro a few days before he showed up in the ER by ambulance one night with a new hemiplegia and a hemispheric bleed on CAT scan. The INR, a measure of clotting, normal 1, therapeutic range 1 to 1.5 to 2.5, intensive 2.5 to 3.5, was 9. He deteriorated in the ER and died in the hospital before the next sunset. Just because it stinks, it doesn't mean it's all bad. So they knocked off his gut flora, no vitamin K from there, and he was getting an antagonist of vitamin K to stop his clotting. So he had issues.
Had a stroke.
Yeah.
Interesting, huh? How everything works together.
Yeah, really interesting.
All right. Now, I had one email come in this week, which is not a follow-up, but I thought I'd read it because this is National Influenza Vaccination Week.
Yes, it is.
Martin writes, I am a 69-year-old in fairly frail health. The VA clinic gave me the high-dose vaccination last month. If I start feeling flu onset, I plan to go to the ER and get an antiviral course. I seem to recall you talking down the effectiveness of Tamiflu against flu. Should I be worried?
The current strains in circulation that the CDC has found so far are susceptible to the antivirals.
Yeah, I remember we were saying it doesn't help all that much because of those meta-analyses.
Right. The meta-analysis suggests maybe it's not that great. On the other hand, the side effects seem to be pretty mild, so, you know.
Not only that, but here's my sort of redux of all of that conversation we had about the meta-analysis, and that was that it was mostly looking at the outcomes from various clinical trials where these things are given to people who are healthy, and then looking at what happens to them. And we got a lot of feedback from doctors who said that in a hospital where you're dealing with people who are really sick, the experience, at least anecdotally, is different than that.
That's right.
Okay. So there may be some effectiveness that is not really covered by the meta-analysis in a situation where you're dealing with people who are really sick. So am I saying all that right?
Yeah, I think that's right. I think that we had a few physicians write in and say, we use it. We think it works.
Yeah.
They said, we know it's not a clinical trial, but seems to be good.
And as Alan says, it doesn't hurt.
Right.
So, Martin, don't worry. If you do feel flu-like symptoms, get to the ER and get antivirals.
You're going to be fine.
You'll be fine.
Everything's going to be okay.
Keep listening. Thank you for listening.
Yes.
All right, so we have two papers which I think you will like, and they were both suggested by more than one person each. Joshua wrote, as fun as the recent Ebola virus binge has been, I hope you find time to talk about sea-star wasting disease. As a layperson, I can't find much info on this virus. Hope you can inform us. You are fantastic teachers, and your listeners, dedicated learners. Thank you. Also, Heather wrote, please, please, please consider the attached paper about sea-star wasting disease. She liked Bulldogs Go Viral and TWiV 310. It almost made me want to get out my old microbial genetics materials for review. And here, listen to this. On another note, I hate to admit it, but I have minutely missed this week in Ebola. I hate to admit it, but minutely is not that much. All right, so the first paper is published in PNAS not too long ago. It is called densovirus, associated with sea star wasting disease and mass mortality. A lot of authors: Hewson, Button, Gudenkout, Miner, Newton, Gados, Wynne, Groves, Hendler, Murray, Fradkin, Breitbart, Fassbender, Lafferty, Kilpatrick, Miner, Raimundi, Lahner, Friedman, Daniels, Helena, Marliev, Burge, Eisenlord, and Harvell. And you know what? Raimundi, that's my middle name.
No kidding.
Yeah, as my grandfather was a Raimundi.
Huh.
Peter Raimundi. I wonder if he's related. There are lots of Raimundis. The Racaniellos are all related.
So is Raimundi an Italian name?
Yeah, it is.
It ends in a vowel.
Yeah.
So that's
Yes.
good.
I got 2 vowels on 2
Wow.
of my names, you know. Yeah. This is from
And
all
these
over.
people, not only there are a lot of authors, but they're from all over the place.
Yes.
Yeah, they are from all over the place. Both coasts, right?
Yeah, both coasts. A lot of them on the West Coast.
Canada?
And aquaria, a couple of aquaria in there, right? Because these sea stars, you know, they have them in aquaria, I presume, right?
Yeah.
Sure.
Because people like to look at them.
Yeah.
And their sea stars are asteroids.
Yep.
So they don't. It's not the same as starfish, right? That's different.
Right.
No, they are starfish. If you look up,
Okay.
They're called.
as a matter of fact, I looked up. I started off by looking up asteroids. This is all Wikipedia, and I wound up with. I'll look this up again. Dang, but it's asteroids include starfish and sea stars.
And they really shouldn't be called starfish because they're not fish,
They're
right?
not fish.
Yeah.
No.
Maybe I figured they're trying to get away from that nomenclature.
So here we go. If you look up starfish on Wikipedia, the classification goes kingdom, Animalia, phylum, Echinodermata, subphylum, Asterzoa, class, Asteroidea. And then there's a whole bunch of orders. And the picture is, sort of a standard starfish. But there's all kinds of others within the class, all bunch of other orders that you can click on, and there's they're amazing, these things.
Yeah, they're beautiful.
They're really
Check 'em out.
amazing.
Yeah, when I was in Vancouver, there were tons all over the rocks. You know, when the tide goes down, they're all different colors, really pretty.
Well, the West Coast where I grew up is a wonderful place for this kind of stuff because they have these tide pools, right, that are just full of all of this stuff. Ooh, wow.
Right. So just on the taxonomy, the Asterozoa is the subphylum, and then there are 2 classes within it. There's Asteroidea, which is the starfish, and Ophioidea, which is the brittle stars, which are much they have thinner arms is the most obvious feature. of
Okay.
those.
So the story here is that there's been a big die-off since the middle of 2013. Millions of these guys have, as they say in the first sentence, have wasted away into slime and ossicle piles.
Yeah.
Very graphic.
This reminds me of what baculoviruses do to caterpillars.
Yeah, that's right.
They melt.
And this has gone from Alaska to Baja, California. Many different species have been affected, and there's some... what happens is they get lesions, and then their limbs start to curl up, and they deflate.
PNAS is open access, is it not?
Depends on whether you
One
pay.
of these was open access, and the other was not.
You have to pay to get open access. Yeah, freely available online through the PNAS open access. So this one, the Sea Star, is open.
Right.
Okay,
The other one is
because
not.
the pictures are cool.
Yeah.
Okay,
Yes.
that show the pathology of this.
Yeah,
thing.
they have
It's
pictures,
amazing.
and they eventually melt and they die. And they say maybe the biggest marine wildlife epizootic ever observed. So lots of these are dying, and all of a sudden, it's not clear that this is happening for a long time. So has been some evidence that it's an infectious process because it can move from site to site and so forth. So what they did here was try and figure out if, in fact, a virus was involved. And
One of the things that caught my attention early on in this, outward signs of sea star wasting disease vary slightly among species, but generally start with behavioral changes, blah, blah, blah. And I'm thinking,
behavior.
How do you know?
how do you
Yeah, it's
know?
pretty funny. Yeah.
Right. But behavioral changes, including lethargy, boy, you got to watch them for a long time to know they're lethargic. and limb curling, followed by lesions, ray autonomy, turgor loss, and end with animal death. But I thought that was cool, behavioral changes.
So they collect some of these sick guys and healthy ones, and they put them in tanks in their aquaria. And then they make filtrates from the sick sea stars. They grind them up and homogenize the tissue, and then they filter it to remove the crap, and then they put it through another filter it's a 0.2 micron pore filter to only let the smaller viruses go through. Of course, that would hold back a lot of the bigger ones.
Yeah, they must have had, clues by then.
And then they inject this into the asteroids. Just pull them out of the tank, inject it with inocula into the coelomic cavity.
The gut.
The gut, right?
0.2 microns is like 20 nanometers. That would even keep poliovirus out, right? They're really focused on little guys at that point.
Yes, these are the smallest ones. That's right. I wonder why. I wonder if they
probably already had some preliminary data by the time they got to that procedure.
They may have. I suspect what they may have done is like a preliminary experiment where you do a 2 micron or a 1 micron filter, and it's still there.
Yeah.
Okay, so it's viral. So let's see what size virus it is, fractionated to 0.2 microns. You get just the tiniest viruses, and now it's still there.
Right.
And it's dramatically there when they put it. I mean, the controls are just fine. And the experimental animals are all dead.
Right. So they transmit this infection by inoculating this into the gut, and it's seems to be virus sized. Of course, it could be not a virus, but I think there's pretty good evidence that it is. And so they ask they look in extracts of diseased sea stars, and they can see particles, icosahedral particles. They're 20.
And they actually did the infection thing 2 cycles.
Yeah.
So they took the ones that had died in their tanks in their experiment, extracted them, and then did it again.
Yeah, as we say in virology, they did 2 passages. Yeah, they see particles in the sick animals. So they say, what's here? So they do deep sequencing, metagenomic analysis. They excise tissue samples and extract nucleic acids. They do both DNA and RNA, and they deep sequence it to see what's there. So they get a bunch of different viruses. They get big DNA viruses like Mimi viruses and phycodnavirus, which is the subject of our next paper. They get some RNA viruses, retroviruses, dicystroviruses. Those are picorna-like viruses with 2 open reading frames, and parvoviruses. So they get a lot of different ones, but all the samples with the parvos were from symptomatic asteroids. So that's the reason for focusing on this, even though they found lots of other viruses.
So they say metaviromic analysis indicated a greater prevalence of parvovirus-like sequences in symptomatic relative to asymptomatic.
Right.
So it's 2 of 13 asymptomatic libraries and 7 of 15 symptomatic. So it's not black and white.
No, it's not.
But there's a clear bias towards symptomatic.
So this parvovirus, they look at the DNA, and it turns out to be a parvovirus of the densovirus. Is that a genus, the denso, or is it higher than a genus?
Oh, man.
Look it up on
I'll look it up.
Viral Zone.
I don't know what that must be. I'll bet you that's a genus because the family is parvovirus. Go to Viral Zone.
Yeah, Viral Zone will do it.
But I looked this up because I didn't know the difference between densoviruses and other guys. And the striking difference is that whereas the other parvoviruses have all of the genes, there's only 2 of them really, off the same strand. The densoviruses are essentially ambisense, though that's kind of a weird term for a DNA virus, but they have the genes in opposite orientations. One's in one orientation, one in the other. And they have bigger ITRs.
So these are single-stranded DNAs. They are small, 25 nanometers. And so now they have probes for this particular densovirus. They call it C-star-associated densovirus. Now when they do an infection, so when they infect healthy stars with extracts of sick stars, they can now monitor virus loads by PCR And they see this virus goes up when you inoculate a healthy animal. And the healthy animal gets sick, and that correlates with an increase in the genome of this virus.
Okay, so Densoviridae is a subfamily.
Subfamily, okay.
The family is Parvoviridae. The subfamily, there are two subfamilies, Parvoviridae and Densoviridae, and in each of those there are a bunch of different genera.
Okay.
Okay.
All right, then they do field surveys now that they have probes for this virus. They go out there and they look in sick and healthy sea stars, and in fact, the virus is more abundant in disease than in healthy, but it's not black and white, as Rich said. Some healthy ones have them, too.
They also kind of speculate that might be because they just haven't gotten sick yet.
Could be.
Because there's about a 2-week incubation period.
Right, they're asymptomatic. It doesn't mean that they're fine.
Personally, I think they ought to quarantine all these sea stars. I just think
at least keep them off the subways.
They should screen them at the airport.
They found the virus in marine sediments. They find it in plankton, and they find it in sea urchins, and also in sea stars that have been preserved in museums since 1942.
The sea urchins are non-asteroid echinoderms. The Strongylocentratus purpuratus is the only one of the echinoderms whose Latin name I know, and that's the purple sea urchin.
Yeah. Yes, I have a long and crazy association with those critters.
I remember you talking about that. Yeah.
There was a guy when I was in college. There was a guy who actually we looked at. They're a great species to look at embryogenesis, or at least the early stages. You can harvest sperm and eggs from those critters and fertilize in vitro and watch the first few cleavages. I've seen that. Seen that happen.
Yeah, Dave Eppel did that at UCSD, and the reason I know it is a friend of mine rotated in his lab, and that's the one thing he could remember was the name of the purple sea urchin. And
yeah. I guess it's not forgettable.
Right.
So here we have these densoviruses. They've been around a while, but the disease is not continuous. So this authors speculate that there must be some reason why we just have had this recent outbreak. And Alan Dove, in fact, had a good idea, which he wrote about on my blog, Alan.
Did I?
Did you remember? Do you remember?
What did I comment on? Oh, yeah, pH tolerance.
Yeah, so maybe, the pH of the oceans are being reduced. Maybe that has something to do with it, or some other environmental factor.
We're doing this big global experiment right now where we gradually reduce the pH of the oceans, and we know corals don't like it.
Right.
And I don't know if it's having an effect on this virus.
So I think what they need to do, and maybe this is hard, of course, is to get the virus in culture. I don't know if you can make cultures from sea stars, but grow the virus and put it in and show that it causes the disease, right?
Well, I wanted to challenge you on that because you said that on the blog that's what they need to do is be able to grow it in cell culture. But we, I think, have lots of viruses that we can't culture in cell culture that we believe are causative of disease.
Tell me one.
HPV.
Norovirus?
Human norovirus. Used to be hepatitis C.
Yeah.
Papilloma.
Well, I agree that the evidence for those is overwhelming at this point. Plus, you can vaccinate against them and prevent infection or use antivirals. But this is the first report, so presumably over the next years, if we get additional evidence. But I think that would be like the gold star. Right?
What if they?
Bet you. I'm sorry. Go ahead, Kelly.
Well, I was going to say, what if they took their second-round infected animals, homogenized them, did their unbiased deep sequencing of them, and only found this virus? Would that be convincing?
I think that'd be pretty good, yeah. But why can't they culture it? I guess it just might not be cultured.
Listen, I'll bet you with this guy, if you they don't have quite the full-length sequence, they don't have the inverted terminal repeats, but I'll bet you if you did, you could just inject DNA into these starfish and they'd get sick. I'll bet.
Yeah, it could be. You guys see that paper by Eric Delwart where they reconstructed I think it was a parvovirus from 700-year-old caribou feces?
I didn't read the paper, but I saw some coverage of that.
Yeah, so they actually put the DNA into, I forgot what the host was, and it replicated. So, yeah, you could do that if you had the whole sequence. I'm sure they could get it. Yeah.
So Eric got tired of blood, and now he's, going for old poop.
Yes. In fact, I had originally put that on the agenda for this episode, but then I thought I should do these instead. I don't know why. They're all cool. But, yeah, I don't, and not in all cases you'll be able to do that, Kathy, but I would say that if you wanted to have it today, just try growing it. Someone actually posted, and I'm maybe they work on this. Where is it? It's not here anymore.
So you want them to develop some kind of cell culture system from one of these sea stars.
Yeah, well, if there isn't one, I understand that it's difficult, but
I mean that could be just a big fishing expedition, as
so
it were.
to speak.
Yeah, but you could try. I just don't know the history of this field. People may have tried and it doesn't work, in which case, okay, that's fine. Try some other host, but apparently has a broad host range, right? They find it in, echinoderms and other things. So anyway, another thing I thought that was interesting here is they found these retrovirus sequences, and they say, here's a quote, retroviral annotations are likely spurious because they were detected in DNA and have RNA as nucleic acids. So I thought, come on. Of course, they're in the DNA. They're integrated, right?
They're retroviruses, yeah.
So they didn't pick that up. So I guess, well, anyway.
There's a 1995 paper describing a method for culturing embryonic cells of a starfish.
I certainly would love to try it, because that's the kind of thing we like doing. And then you could make a plaque assay.
Right?
You're optimistic that all these things are going to work, but
of course. Well, you got to be an optimist, right?
Yeah.
Okay, so that's that. Anybody have anything else they want to say?
I did want to say one thing, and that is that they speculate on some of these things, like the sea urchins and so forth, that the impact of the virus on them isn't known, and maybe they could form a reservoir. And here we go for This Week in Vocabulary. I love this. They said, if these are tolerant reservoirs of infection, it might help keep the virus in the system long after the virus extirpates less tolerant hosts, helping explain how SSWD, the disease itself, can extirpate some host species. So extirpate is such a cool word. I only know of it from Elijah by Mendelssohn, where there's a chorus, Baal, let thy flames fall and extirpate the foe. So a plug for music informing my science vocabulary.
Yeah, extirpate is a good word.
Yeah.
Root out and destroy completely. That is a great word.
Yeah.
All right. Now, the next paper was recommended by Robert, Alberto, Peter, John, Harold. Yes. So this really got people excited. Let's see if there's anything cool. Robert had a lot of questions about it. What is the virus? How is it transmitted? What areas of the world? I want to know what we think about it. A headline, too good not to forward the link to you. Algae virus found in humans really make them dumb, This is Peter from Korea who just wrote above. He wrote 3 times now. And then John wrote about it. John is our friend here in New York City. Okay, so the paper is... Oh, this is a cool thing. Long-time listener, temperature in my office, cubicle, 79.2 degrees F. Don't have a view out a window, but all fluorescent overhead lights are functioning. It's great.
That's good.
Yeah.
The paper is also in PNAS, and this one is not open access. It's called Chlorovirus ATCV-1, is part of the human oropharyngeal virome and is associated with changes in cognitive functions in humans and mice. The authors here are Yolkin, Jones-Brando, Dunnigan, Cannon, Dickerson, Severance, Sabuncian, Talbot, Prandovsky, Gernon, Agarkova, Lester, Gresset, Chen, Deuber, Ma, Pletnikoff, and James Van Etten, a.k.a. Yoda. I was at a meeting.
Since when is Jim Yoda?
Well, here's the story. I was at this giant virus meeting in Germany a year or two ago. I was doing a podcast with Jim and Michel Claverie and Chantal Albergel. And I introduced them, and I said to them, would it be okay if I if I called Jim the godfather of giant viruses, and someone in the audience said, Yoda! And I looked at him, and he does look like Yoda. He's got his bald head and his hair, wispy hair sticking up, and anyway, he's a good guy. So Jim has been working on these chloroviruses for years. They were the first really big viruses, and now, of course, they're eclipsed by the Mimis and the Pandoras, etc. But he still keeps up. And when I visited him in Nebraska some time ago, he hinted at this work, because I actually asked him, hey, do they replicate in HeLa cells? And he said, oh, I'll let you know. So in this study, I'm not sure why they did this, but they had a study of adults, for which samples were collected for other purposes. 33 adults without a known psychiatric disorder or physical illness. And they had oropharyngeal swabs, basically. And these were, as I said, collected for some other study. And so they looked by metagenomic sequencing to see what was in there. And they found lots of hits for chloroviruses, which is a large double-stranded DNA virus of the phycodnaviridae family.
So these things have an interesting
Condit,
biology.
they're not things. Come on.
I'm sorry. Oh, there's a picture of Jim. Hi, Jim. So they have an interesting biology. They replicate in blue-green algae, right, which are photosynthetic algae. But these algae have a symbiotic relationship with paramecia. Okay, and you can find free virus in pond water all over the place at fairly high titers, but the infection actually only takes place in the context of the blue-green algae that are inside the paramecia. And if you look up online for the paramecium or the blue-green algae, you can see pictures of these things that are these protozoans, the paramecia, that are just chock full of these blue-green algae.
Yep, it's pretty neat. Anyway, so they had done this. These samples were collected as part of a study of cognitive function. Now, how Jim got a hold of these or why, I have no idea. Maybe he'll tell us.
I would imagine that, what makes sense is that they got these sequences and said, what?
Yeah, maybe they got it that way. Yeah, that could very well be. Because Bob Yolkin is in Baltimore, which is where these sequences were taken from. So anyway, they found chloroviruses in 40 of 92 individuals, 43%. And that was because it was part of this study of cognitive function. They had done tests on these people of various sorts. They put them through mazes. No, sorry. They could figure that the presence of DNA was associated very slightly with
very slightly
decreased performance in tests for visual motor speed, delayed memory, and attention, all of which I exhibit substantially.
And we should probably note this group of people was not geographically random. This was a group of people from Baltimore.
Baltimore, yep.
As the Hopkins group.
Which is probably not a good thing, because...
Probably not the best. If you've seen John Waters' films, you know that some strange stuff is in the water down there.
Edgar Allan Poe.
Yeah.
Wasn't he from Baltimore?
Yeah, a lot of his life he lived there.
So
H.L. Mencken.
Well, you grew up not too far from there.
Yes, I did.
So, all right, that's the story. And I asked Eric Delwart, and he's like, I'm worried about contamination because the lab works on these things. I'm worried about columns that may have chloroviruses in them and so forth. So he said, I'm a little worried about this. But then what they do is they do an experiment. They feed mice chlorovirus-infected algae, right? And then the mice, they measure them for tests of cognitive function. So they ask them questions, I suppose.
They take them to the opera.
Recognition, memory, sensory motor gating. And they have decreased performance, and they also develop antibodies against the virus.
Some of them do.
Some of them do, and then they look at... They do this experiment where they basically look at gene expression in the hippocampus, which is the part of the brain that would be involved in these functions that they measured, and they find gene expression changes there, which is a huge stretch to say that it has anything to do with the virus. So that's basically the story. And I just don't know. These viruses are present in inland waters, right? They're very common. And you can imagine, Baltimore's got water around it, right? Or no, Alan?
Oh, yeah. Lots and lots of water.
Yeah, so these people...
lots and lots of algae.
So people could be inhaling these on a regular basis, and that may be why...
They could be inhaling them. They could be in the oysters. They could be in the crabs.
The what? The oysters?
The oysters. You get some half-shell oysters.
I thought you were saying something scientific.
No.
Yeah. So I just don't know if it's the virus that's doing anything, or if it's... You don't even know if it's replicating in them. So I'd like to know if this virus could replicate in
humans.
We don't know. That's an important question.
They did antibody titers on the mice. They didn't do antibody titers on the people, right?
I don't believe so. They did not, right? Because that's something.
And it's a small, geographically localized sample. And looking at these graphs, I'm not a statistician, but the error bars are ginormous.
Yeah.
And that's a function
the differences
of
are small.
The sample size. So the mouse data are a little maybe cleaner, but still, it's not. It's not a huge effect.
So someone wrote on my blog, I think this is quite. Far more likely scenario, the chloroviruses are coming from environmental contamination. The mouse studies are bollocks. That's what you get in blogs. But I just think they need to do more work. So the questions that people sent in, is this possible that this is doing this? And I think it's too early to tell.
Yeah, I agree.
So, Alan, this was attractive for the headlines because the virus makes you stupid, right?
Total headline material. I mean, you can't not cover a story about this with anything that's got a link to human intelligence or human behavior, and
people would like to blame their issues on other things like viruses, right?
Of course, and and it's just way too easy to do a story on, is a virus making us all stupid or making half of us stupid, and how would we know which half? And another issue with this is, besides being one sample of 92 people, as far as I can tell, this was at one point in time.
Yes.
So we have no idea if this is something that persists. Is this part of the normal human pharyngeal virome? Or is it part of some food product that's very commonly consumed, or some activity that's very commonly done in the Baltimore area? That exposes people to it temporarily, has this effect, and then goes away.
Well, they put in the title, the virus is part of the human oropharyngeal virome. I don't think that's quite possible to say yet.
Yeah, we don't really know that for sure. We don't really know that the behavioral effect is real.
Yeah. So intriguing, but needs more work.
It's a neat. Obviously, they took a sample of opportunity and did something interesting with it, and found something tantalizing. And this is something that is worth following up on.
And my guess is, if you asked any of them if they had a lock on this, they'd say exactly the same thing.
Probably.
It needs more work. But it's the kind of thing that's it's interesting and crazy enough so that there's no reason not to publish it at this point. It's not It's not obviously dead wrong or anything like that. It just needs more work, and it's tantalizing.
Unfortunately, a lot of the news coverage on it implied a much greater degree of certainty than the authors would endorse, I'm sure.
Sure.
So Eric Delwart says, it's certainly feasible that people would have ACTV in the back of their throat, but I'm skeptical this would impact visual processing. Could it account for why people who live on the seashore have a more relaxed outlook in general?
Yeah, except that these aren't really saltwater critters.
No, they're not.
We're talking freshwater.
They're freshwater.
So, well, the bay is
it may explain
so
Walden Pond.
Get your choice.
Yeah. So he said the hits look real. They don't look like contamination, although he was worried. But he doesn't know how they... He said, ask Ian Lipkin, because he's a neurovirologist.
Well, the mouse studies are really an important part of this. Okay, if without the mouse studies, you might really shrug. It's just an association, and there are so many other variables that could make them seem to be associated. But the mouse studies kind of give it a little more credibility. By the way, I put it in here that I found a link to various intelligence tests for mice because those always fascinate me. And there's a Stanford site that summarizes a bunch of these. They're interesting.
Hey, Rich, you remember the paper we did where they look at how mice react to something? I can't remember which. And the picture in the paper were all these different expressions. All the mice look the same.
That's right.
Puzzlement, depression, this, that. They're all the same because mice don't have symptoms, right?
Yeah, this was a paper that had to do with cocaine addiction in mice, and I forget quite the deal, but one of the behavioral things they had was simply a camera over the cage, right. And they could trace the path that the mice were taking, and they had diagrams of how crazy the mice were based on how they ran around.
Those are the open field tests, I think, and in fact, these authors use that too.
Yes.
And in that one, mice mostly will stick around the edges of the cage, and if they venture out into the middle, that's an aberrant behavior. It's a suppression of defensive behavior. They want to hang around at the edges, and if they venture out in the middle too much, then that's unusual.
I was talking with Glenn Rawle, who's an RNA virologist in Philadelphia and a co-author on the textbook, and he told me about work with lymphocytic chorial meningitis virus, which does affect mouse intelligence. So they do these maze tests where they train a mouse, they put a mouse in a maze and there's a fork. They put food in one end of the fork, right? And the mouse quickly learns where it is. Then you change it, and normal mice learn 2 or 3 tries, they learn where it is. But if you infect them with LCMV, then they get stupid. They can't figure it out for many, many times. Stuff like that. So is a precedent for this. It does happen.
Yeah, that Y maze is one of the tests they used here, and that the ones exposed to the virus didn't do as well on that Y maze test.
I don't know. If they put me in a Y maze, I don't know if I would do very well either.
Depends on who was asking why.
Ooh. Let's do some email. What do you think?
Sure.
Russ writes, Vince and friends, I think you missed the point of Alice's very short post. It is a haiku. Love them, but they are so long, I just don't have time to listen. 3 lines, 15 syllables, 17 in classic Japanese haiku. And he gives a link for that. What do you think, guys?
I think that's brilliant.
That's wonderful.
Mm-hmm. We missed that one completely, and we just gave Alice all sorts of trouble
for
saying that they were so long, when in fact she was being quite clever.
Yes.
Sorry, Alice.
All right, sorry, Alice.
Go ask
Our
Alice.
Apologies.
Also, read over my letter about airborne versus droplet. You still get them confused. Airborne spread by small airborne droplet nuclei. They remain suspended. Droplets spread by large droplets. I don't think we get it confused.
I don't think we get it confused.
I think that's exactly what we say, right, guys?
Yeah.
Okay. Alan.
Marianne writes, Hi, TWiV hosts. I couldn't help but chuckle to myself when I heard you read Dave's email regarding the confusion of receiving so many emails from Dave's. Reminded me of a great comedy sketch from the classic Canadian comedy troupe, The Kids in the Hall. It's below. It's called Dave's I Know. And sends a YouTube link. Isn't there also a Monty Python sketch with the Bruces in Australia?
No, I don't know.
I don't know.
But I always think, and I think this was your title idea of the Seuss book, Too Many Daves.
Yes, there's a Monty Python sketch. Bruce.
Rich Condit. Can you take the next one?
Pat writes, Dear Doctors, thank you for your excellent podcast. TWiP.
Yeah.
TWiP 76. Okay, herpes virus worms its way out was fascinating and led me to wonder whether pre-existing parasite infections may be one of the reasons why Ebola has a higher death rate for residents of tropical areas of Africa where parasite infections are more common than for residents of the United States or Europe so far in these very few cases. In addition to the links you provided, this paper in Science looks at the effect of parasite infections on viruses, and she gives a link. Interestingly, a sort of editorial paragraph has been added titled Parasites Make It Hard to Fight Viruses. Do you happen to know if this interaction is being specifically studied with regard to Ebola and whether addressing chronic health issues might help people be more resistant to Ebola and less likely to die from it? Thanks.
And I asked Dixon, and he shrugged.
I think addressing chronic poverty issues might help people be more resistant.
Yeah,
to
I
Ebola.
think the biggest issue with the different mortality rates in Africa and here is just the quality of the health care that people get. I mean, it's not unreasonable to think that there could be an interaction between the virus and parasitic infections, but my guess is that that's far from the major thing that's going on here.
I don't think you need to be so these parasites can immunosuppress you, and I don't think you need to be immunosuppressed to get it wiped out by Ebola. So I looked, and I didn't find anything where they try and look at the effect of these infections on Ebola.
Well, here again, it's the kind of thing that there haven't been enough infections of Ebola in the past to make this a line of research to do.
Precisely. Exactly. All right, I'm going to take the next one, Kathy, because it has to do with polio. Then you can take Bill's, all right?
Okay.
Sam writes, Dear TWiV, I saw this on Science Daily, a paper in PNAS describing vaccine resistance in a polio outbreak from 2010 in the Democratic Republic of Congo. A few questions. It seems this strain was isolated during an outbreak. Is it possible this mutation was wiped out with the outbreak? Could the mutation be overcome with more doses of vaccine, or would a new version of the vaccine be needed? Thank you for your perspective. Weather here in Tucson is clear and sunny as usual. High of 71, low of 51, humidity 33%. Bit of a cold snap, but we'll be back in the 80s later this week. So I wrote about this because it's quite interesting. It was an unusual strain of polio, which is quite resistant to neutralizing antibodies, isolated in 2010 in the DRC. And it arose probably because the immunization had dropped and there was virus circulation. But they went back in and immunized, and it wiped this out. So it has fitness costs associated with this change in the capsid, which makes it relatively resistant to antibodies. But they did some really cool studies in the paper where they showed that basically, if you take sera from people who are immunized, it barely reacts with this virus. So it's a good thing it's not circulating. But if you have a well-immunized population, it's really not going anywhere. So that's why it's so rare. It's only been isolated one other time in Finland. And if you keep immunization levels high, you don't see this. So that's the story there, Sam, and I'll put a link to my blog post where I discuss that. Okay, Kathy, take Bill's, please.
Sure. He writes, Thanks for the entertaining and informative program. Last week, I thought I understood someone to say you feel obligated to respond to all emails. Well, feel free to ignore this one if the question has already been answered.
Okay, next.
I read the CDC recommendations for care of Ebola virus disease patients in hospitals, including Bellevue, NIH, etc. They seem clear for anyone in the facility potentially exposed, down to developing sick leave policies. My question regards workers who don't feel they've been potentially exposed. They don't live at NIH, so I assume some come to and from work on public transportation, eat out, go to PTA meetings, etc. I can see no reason not to, as long as they are asymptomatic. Given the near panic raised by one well nurse in rural Maine, perhaps it is better not to discuss all the Bellevue workers moving anonymously among us anywhere near governors or media. How is this actually handled? I am a retired clinical psychologist with no background in virology or communicable disease, but taught interns in family medicine how to interview without falling out of their chairs for a while. I had the privilege to attend 8 years of noon conferences on many issues. There was an epidemiologist on the faculty who repeatedly urged residents to ask patients if they had traveled recently, what kind of work they did for potential exposures, and to consider educating themselves about the environmental factors affecting their patient populations. He was generally ignored by most residents, but must feel thoroughly vindicated today. By the way, I'm not aware of any of our residents practicing emergency medicine in the Dallas area. Sorry to be so long, but I've always admired writers who could communicate scientific ideas to the general population, Asimov, Gould, etc. I was wondering if you had any comment on the book that caught my attention a year or two ago and that I recently reread, Spillover by David Quammen. If nothing else, his narrative accentuated the role of veterinary scientists in field and lab research of emerging diseases. Thanks again, Bill.
Yeah, what does this mean, falling out of the chairs? Do you understand that?
Oh, that you shouldn't react. Probably.
Oh, okay.
You just want to be kind of stone-faced and taking down the information.
Okay.
When they say something absurd or
Right.
scary or whatever.
Or hilarious.
Yeah.
My closest, association with physicians on a regular basis has been with infectious disease docs at a conference that we have here on a monthly basis where they go over a couple of cases. And for the ID docs, asking, where people have been and what their circumstances are and what they do is standard operating procedure for a history.
Right.
Yeah.
Here in New York, obviously a lot of people went in and out of Bellevue, but they don't talk about it. And it's not an issue because unless you can get it from a sick person, right? But if they're exposed and they're not sick, they're not going to transmit it to anyone. That's as far as we can tell. So that's why they let people move around, and now it's over here.
Yeah, it's over. That whole thing has really died down.
This book, Spillover, is very nice. I read it a while ago, and David Quammen was at ASV last year, American Society for Virology. And I talked to him a bit, and he's a really nice guy and would love to come on TWiV and talk about his work. He said that the best way to write about science is to write about the people who do it, because that's what people find interesting. And in his book, he talks about a lot of the veterinary scientists who figured out some of these spillover virus infections. So I like it. There were a few mistakes, but overall it was good.
I thought it was very good and very thoroughly researched. The guy really
He was very good. He understands what's going on. There was a section where he was traveling through the forest somewhere, and he kept talking about drinking warm beer and eating this stuff, and I found that a little tedious. It wasn't really very interesting to me personally, but I could see where a lot of people might like that. So it's good. He just is revising it, I think, to update it, obviously, for obvious reasons.
Yeah.
Okay, Sandra writes, is it known if some body fluids have more Ebola virus than others, particularly wondering if nasal and or lung secretions have lower levels of virus than blood, vomit, diarrhea, saliva? Oh, I don't know if we know this because not so many studies have been done, right? I
don't have an answer to that.
There's that one paper that we've talked about on TWiV before in Journal of Virology. Darn, I was going to look at it before the episode.
It used to be right in my papers, and I recently moved it out of the inbox.
You remember it was a Jonathan Towner CDC study, right?
Right.
Let me see if I can find it. What would it have been? What episode?
Oh, so it's the Towner paper, Journal of Virology, yes. April 2004. Assessment of patient viral load as a predictor of outcome.
Yeah, but I think that's just serum, though. That's not other body fluids.
Oh.
I don't know, Sandra. Does anyone know?
I'm working on it.
Are you working on it?
Yeah.
You want to work on it? We'll do the next email?
Sure.
Alan,
I'll just check out here.
Yeah.
Okay.
You're always checked out.
All right. Trevor writes, hello to my favorite virology podcast. And by the spelling of favorite, I gather this is a Brit. Oh no. I'm a Canadian university student who's interested in virology, specifically the use of viruses as vectors and RNA interference, which silences or knocks down genes. I've been in correspondence with several professors at my university, but unfortunately most of them don't have the answers to the questions I have on the subject. And since this is a somewhat new technology, I'm having trouble finding places to research it. Do you know of any individuals who have done extensive work in this area or are at the cutting edge of this field? I have so many questions about why the effects of RNA interference are transient and how they might be used in the future. So if you know of any good texts or papers on it, I would be grateful. As always, your work on the podcast is excellent, and I think you and your cohorts have gone a long way to educating your listeners not just about virology, but how to think critically and creatively about the field of science in general. By the way, the weather here is about 12 degrees Celsius and sunny.
I would say, we've had a few people here on TWiV, Ben Tenhoover and Chris Sullivan, who do RNA interference, and they produce them in vectors and look at what they're doing. The guy at Duke, Brian Cullen, is also another leader in that field. So I think, Trevor, if you go back, you can find some of those old podcasts and listen to them talk about. There'll be links in those, right?
Yeah.
How you doing, Rich?
I've got, is this the paper you were talking? I've got a paper from Journal of Infectious Diseases. Towner is on it. but its first author is Bausch, and the last author is Rollins.
Yeah,
it's got
that
a
might be the
table
one.
in it that talks about number of percent PCR positive samples in acute or convalescent individuals, and compares saliva, skin, urine, vomit, sputum, breast milk, stool, sweat, tears, nasal, blah, blah, blah.
Right.
So here we've got 8 out of 12. I'm looking at the acutes. 8 out of 12 in saliva, 1 out of 8 on skin, nothing in urine, nothing in vomit, nothing in sputum, 1 out of 1 in breast milk, 2 out of 4 in stool, 1 out of 1 in tears, nasal blood, 1 out of 2 in semen.
So don't bring that up.
Yeah, sorry. That's not. It's not a huge sample size.
It's not quantitative either,
and it's not really quantitative.
I'm not sure it's been done, but
I have fields open on my desk because I'm working on a paper for a class for that. I'll see if I can find something.
All right, let's do one more email. Hey, Rich, you up for doing?
Sure. If I can find out where we are.
Christoph
writes, "Hi, Vincent et al. Ebola." Just kidding. This is not about Ebola virus. I was having a discussion at home with a family about the flu shot just now. The whole family got it the other day when we went grocery shopping, as the pharmacy was doing free shots. Huzzah for the Canadian health system. I was joking about it being so easy I could go and get a flu shot every weekend when we do the shopping. So, not that I would actually do this, but I'm curious. If you don't have a negative reaction from your first shot of a specific year's flu vaccine, would each successive shot of the same vaccine increase the risk of a negative reaction, using up to 1 more of your 1 in X chance of an adverse reaction? Or if you don't have an adverse reaction to it, will it never have one? Or is it that each injection is still 1 in X chance of adverse effect each time you take it? Are there other possible ill effects from having multiple doses of a flu vaccine? Are there any potential benefits from having multiple shots? Thanks for the great podcast. Whoa.
So an adverse event like soreness at the injection site, a little fever, those are telling you that your immune system is responding to the vaccine. It's good. And that would be it would not be influenced by multiple injections, in my view. Right.
I think that's right, because that's an innate immune response. It's not an adaptive response. So it's going to fire every time.
Right. Are there other possible ill effects from having multiple doses? So the only thing that comes to mind is this thing called original antigenic sin, where sometimes when you have multiple doses, you get a memory response to an older vaccine, depending on the strains and so forth, and it may not be protective against the current one. So in that sense, it's an ill effect because you wouldn't have sufficient protection against the current circulating strains. But I'm not aware of any other adverse events that accrue by having multiple doses.
And that's not really a unique problem of multiple doses.
No, not at all.
So if you're getting multiple doses of the same flu vaccine, I don't think it's going to have any particularly good effects. People have looked at, like, what if you give 2 doses? I don't think anybody's looked at much more than that. But it doesn't seem to benefit. I mean, otherwise they would try and go to a 2-dose regimen or something like that.
The fact is that the vaccine pretty much changes in some way every year.
Yes.
Some component of it changes, and so you really need to keep up with the changing strains. So that's one reason why we need it every year. And of course, also the immunity, the memory wanes, so you need to have it every year anyway.
Yeah, and in fact, even if it hasn't changed, you should get it again, just as a booster.
Yep.
But no, every weekend I don't think would boost you any higher.
All right.
A report back from fields. I don't really find anything, but maybe some of our Ebola virus research experts could tell us whether there's higher viral loads in certain body fluids.
Okay.
Yeah.
All right. Let us do some science picks of the week. Alan, what do you have?
I have the winners of a film competition, and I know Kathy's going to do something similar here. So this is a competition for elementary and high school kids, or what are they called in Britain? Primary school and secondary school. So this is in the UK. Or is it just the UK? I think it's just the UK. And they had to do videos about scientific subjects. So you've got the elementary school kid ones are pretty cute. And the secondary school kids actually did some pretty good stuff too. Mm-hmm. And you can flip through these and see who won which one.
It's cool.
They look pretty
videos.
cool.
They're all like 3 minutes long.
Yeah.
Uh-huh.
So one of the judges is Professor Stephen Curry, who
Yes, indeed.
I happen to know.
That's how I found out about this.
Yeah, he's on Twitter, and he's also a structural biologist who used to do some work on picornaviruses. He worked with Jim Hogle.
Yes,
At Harvard. You must know him. You must remember him from your work.
I met him at Gordon Conference.
Ah, very good. Kathy, what do you have?
Well, I have Dance Your PhD winners, and I was just noticing that on TWiV 200, Alan picked Dance Your PhD, and I'm not going to click on it in fear of the fact that it might be an automatic play YouTube.
I think it
but
may
this
be.
one is for this year. The overall winner is a biologist, plant biology student at the University of Georgia, Uma Nagendra, and she and her trapeze class or group do her thesis. And oftentimes there are dances that are supposed to depict certain things, and I just don't get it. But this one was really clear, and it's well worth a look at this, just because the trapeze stuff is neat, but it also portrays her thesis, and there are little captions throughout. Maybe that's what helped me get it. And then if you go further down on the page, there's the winner in chemistry, which was for his PhD dance about mayonnaise, Soya Alvarez. Another guy's dance in physics about nuclear fusion, and another guy's dance about colonialism in social science. And then there was an online vote for a dance of the drones. So a lot of things to look at where people are dancing their PhDs.
Cool.
Why does YouTube have to autoplay? Do we know?
I don't know.
Do they think it just engages people more to do that?
Well, it does have the effect that when I'm looking things up online and I open a bunch of things in tabs, I do immediately get tabbing over till I get to the YouTube video because I've got to find the darn thing and close it. No, I don't want that yet.
There are probably some plugins, there's extensions that prevent that, right?
There may be.
Go ahead, Pat.
In fact, I thought Alan picked one.
I think you did.
Did
Alan.
I?
That's right.
You
Yes.
did.
Oh, somehow I don't seem to have it. Actually, I have something going on in my Firefox browser right now that apparently Flash is out of date, so I don't get an autoplay video. I get this plugin is vulnerable and should be updated.
Yeah, that's a good way to prevent the
That's a
autoplay.
good solution.
Don't
Yeah.
update your Flash. Actually, I think in Chrome there is a plugin that will not play a video unless you click on it. But it has to be a Flash-based video, so I don't know if that works. Rich, what do you have?
I have a science fiction novel that was recommended to me by my son. It's called The Martian by Andy Weir. And it's basically about an astronaut who gets stranded on Mars during a mission, okay? And he's left there alone, and how he copes and the efforts to rescue him. I haven't actually finished it yet, but I'm probably 2/3 the way through it, and I can recommend it with confidence. It's kind of it's written by a guy who's a software engineer. He's pretty young, but he's a self-described science and space nerd, and he's gotten very, very technical in this whole thing. He's got all kinds of stuff worked out, and it's kind of Robinson Crusoe on Mars. It's really a fun read. I enjoy it.
I know why your son is high on this, because all the tech podcasts I listen to, they're all crazy about this. You know, they're all talking about it because he's a programmer. Yeah. So is it gripping?
It's engaging.
Okay.
I don't know about gripping, but it's certainly engaging. I haven't read a book that kept me engaged to this extent for a long time. It's really nice. It's good.
Certainly cheap enough.
I got it on my. I have a sort of ersatz Kindle on my PC for $3.
Ersatz Kindle. Very nice. Is that like a bootleg Kindle or something?
No, actually, you can download Kindle software onto your laptop from Amazon if you don't actually have a Kindle.
It's a kind of Kindle.
Kindle. My pick is Time Magazine this week. Their person of the year are the Ebola fighters. Right.
This is great.
I'm not sure.
The little video that goes with this is fabulous.
Yes, and
it's well worth the 10 minutes it takes.
This, I think there are different people on the covers depending on, where you buy it and so forth. Because I've seen 2 different ones so far, and this I think makes perfect sense. These people are doing important work over there. They're risking their lives. And by the way, we didn't have an Ebola update from you, Rich.
I can give it to you. I got it right here.
Yeah, let me just finish up these picks. We got 2 listener picks. By the way, the scientists or science often makes the cover, the person of the year. The last one I remember is David Ho, who is an HIV guy here in New York, and he made person of the year. And of course, Jonas Salk made it, and other people as well. Our listener picks, we have 2. One from Ricardo. Hello, TWiV team. Greetings from Portugal. Hope Dixon enjoyed Lisbon. I think he did very much. Just a quick reference to new equipment to our home lab. Best regards, and please keep going on your education quest, Ricardo. So this is a Kickstarter that basically is a what is it, an electrophoresis apparatus?
It's a mini PCR machine.
Is it
the
mini
electrophoresis
PCR?
is done separately. I looked into this.
Okay.
So
mini PCR,
this is
righto.
as small as, they got a picture of it here. You can hold it in your hand. It's called mini PCR. If you Google mini PCR, you'll come up with this.
Yeah, he sends a link to it.
And basically, it's a little handheld PCR machine. And my understanding from reading their material is that it does the PCR. You give it DNA, and it does the PCR for you, and you have to do the electrophoresis and analysis of the products independently.
Okay.
But they had I also found the article that describes the Kickstarter to raise money to develop this thing, and basically they were looking for $20,000 and got $300,000, if I read this correctly.
So is this thing supposed to be vastly easier to use than a traditional thermal cycler? Is that the concept?
I think the concept is that it's cheap and small.
Yeah, but you can buy a surplus thermal cycler that's outdated. I mean, you could pick those up pretty cheaply. They're not small, but
how much does this cost?
Like $100 or something like that.
That's pretty cheap.
That's very cheap.
It may be more than that, but that's what I kind of read this quickly. That's
their target price.
Yeah.
Okay.
Well, they're also saying if you have a lab, you should get these because then you can have your own on your bench.
Yeah.
2 by 5 inches, less than 1 pound. Cool. Very neat. Kickstarter supported. And also Eli writes, Dear TWiV team, I have been a Twix fan for the last couple of years. I have a listener pick to propose. Okay, admitted actually 2. It will make me really happy to get them shared on. First one is Battle Ebola with your computer. There's been a long tradition of common people donating computer resources to a wealth of different science projects by downloading a small program called Boink. Large computational tasks get cut down into small bits and sent to participants all over the world. Then your computer can crack away at solving world problems when you leave your machine idle. Buying access to supercomputers can still be quite expensive for scientists, so donating computing time greatly speeds up what research and data crunching is possible. So there's a Boink project related to Ebola, because there are lots of different projects that can be done. And this is organized by Erica Ullman Sapphire. She's probably doing some structural stuff, I would guess, because that's what she does.
There she is on this video.
Yeah, she's on a video, and she's actually it's very interesting. So when I was out in La Jolla, I heard a radio interview with her. She kickstarted some piece of equipment for her lab, which she couldn't buy otherwise for some reason, and was able to buy it.
Cool.
So she was talking about it. So I told her we'd have to get her on TWiV sometime to talk about that and other things. All right, so that's the first one. And then he said, also, I want to share this fine video by Kurzgesagt, which means, in a nutshell, I believe. It's the best educational video I have seen on Ebola.
Hey, come on. Kurzgesagt.
Kurzgesagt?
Kurzgesagt.
Do you speak German?
Yeah.
Hardly. But yes,
Yeah.
I was educated. I was educated. Kurz is small or short. Short. Kurz is small, and gesagt is said.
Okay. Thank you, sir.
Wortbildung. The Germans are famous for building words out of other words.
Hey, I know turducken. That's not German. Wait, I meant to say the Schmutzdecke.
Yes.
Schmutzdecke.
I like the schmutz. Turducken is a fake word, right? Animation style, factual, and with a dash of humor. Thanks for all the good work you do. This is a cute little video by a company, Kurzgesagt, and they do all kinds of different things. And they're animators and journalists, apparently. I'm very jealous of these videos. I'd love to have this kind of animation and to do what I do. There's just one factual error in their Ebola video. They say the cell dissolves and the viruses get out, but we all know that the viruses get out by budding.
Nice animation.
But the animations are cool. They just need a guy with more passion. He's just really factual and straightforward, and he sounds like an announcer. They need a virologist who really feels about these viruses, and that would really clinch it for me. Anyway, thank you, Eli, and that will do it for TWiV 314. Oh, wait, let's hear.
Aren't you going to get the Ebola update?
Yeah.
Wait, and before you do that, turducken isn't a fake word. It's a made-up word, and it's the same kind of genre as what Rich was describing for German.
It's made up. That's
it's a real word.
That's what I meant.
Yeah.
It's made up. Sometimes
but it's in use now, and I'm sure it's in lots of dictionaries.
Yeah, that's the way the language goes, right? We make up words, and then we use them. Okay. Rich Condit, bring us up to date on Ebola.
From the WHO SITREP, as of December 10th, we have 17,942 cases and 6,388 deaths. And, their, what do they call it, roadmap is still being implemented. It looks like in Guinea, the cases are... They say they may be slightly still increasing. It looks to me like it's at least leveled off and maybe even decreasing, but it looks reasonably much better off than it has been. In Liberia, the case count continues to fall and is, for the last week, below 50 new cases. And in Sierra Leone, where the biggest problem is, it is still increasing. But it's slowly, it seems to me, I'm fairly optimistic, being brought under control, though, in Sierra Leone. They still have a big problem, and it seems to me, looking over these charts, that the problem is proportional to the effectiveness or the ability to implement these measures in the SITREP. That is having, in the roadmap, that is having enough beds operational and enough burial teams in place to do this right. So beds and proper burials are the important parameters, and the better off you are with that, the better off you are with the disease, and they're slowly bringing it under control, it seems to me.
Yeah, I was just yesterday or the day before on NPR about a geographical place where, in fact, the Ebola cases are really bad. And I thought that was in Sierra Leone, but I can't be sure.
There's if I haven't read this report in detail, but there are subtleties here with Ebola spilling over into areas that have been naive so far and trying to chase that down and implement the measures there. So they are playing whack-a-mole.
Yeah.
What's that, Rich? Whack-a-mole?
Whack-a-mole?
I know what it is, but I want you to explain it.
Oh, jeez. That's just, it's like an arcade game, right?
Yeah.
With a platform with a bunch of holes in it.
Right.
And these little critters, moles or whatever, poke their heads up, and you try and whack them with a hammer, and they poke their heads up all over the place. And the idea is, as extended as a metaphor, you may be able to keep something down in one place and it pops up somewhere else. So you're constantly playing whack-a-mole. How'd I do?
That was great. I knew you would nail it. It's just great. I've played that game, yeah. All right, TWiV 314. Anyone else?
He pauses.
I'm actually a big fan of whack-a-mole. I almost always win.
Does that apply to work situations as well?
Not so much, but in the actual carnival game, I do great.
No kidding. I don't think I've ever actually played.
It's fun.
You can find TWiV at iTunes and TWiV.tv, and we love to get your questions and comments. You can send them to TWiV at TWiV.tv. Alan Dove is at turbidplaque.com and also on Twitter. Thank you, Alan.
Always a pleasure.
Kathy Spindler is at the University of Michigan in the fair town of Ann Arbor. Thank you, Kathy.
Thank you. This was fun.
And Rich Condit is at the University of Florida in Gainesville. Thank you, Rich.
Sure enough.
I'm not sure if it's a fair town, right?
Oh, yeah. It's a fair town.
It is fair?
Yeah.
It's fair to good, I think.
Yeah.
Have you been there? Yeah, you were, Alan, right?
I was, yes.
And Kathy, I presume you've been?
Long time ago.
Really?
Mm-hmm.
In a galaxy far away.
Yeah. Ed Seiden was still there, so that's how long ago it was.
Yeah.
This is the kind of place where people come for a little while and never leave.
I've been there 3 times.
You're not here forever so far.
No, I'd like to come back and have Satchel's Pizza, though.
Okay, you're on.
You don't have to pay for me. I'd like to get there sometime, and
I'll buy you a pizza.
I really enjoyed it. Yeah, if I get there, you'll buy me a pizza, right?
Geez, I haven't been to Satchel's forever. I need to do that. You're making me hungry.
Okay, you can go. You can go tonight.
I'm going to go sing at a party tonight.
You do?
Yeah. Pediatrics. Good. The Flemtones are really busy during the Christmas season.
Flemtones. That's wonderful.
The fabulous Flemtone.
The fabulous Flemtones, right.
All right, enjoy that.
I will.
I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.